In tonight's lecture, we are going to discuss trait inheritance. Heredity is the transmission of traits from one generation to the next. Genetics, the scientific study of heredity, began in the 1860s when an Augustinian monk named Gregor Mendel deduced its fundamental principles by breeding garden pea plants. Mendel lived and worked in an abbey in Brunn, Austria, which is now in the Czech Republic. Strongly influenced by his study of physics, mathematics, and chemistry at the University of Vienna, his research was both experimentally and mathematically rigorous, and these qualities were largely responsible for his success. In a paper published in 1866, Mendel correctly argued that parents pass on to their offspring discrete, quote-unquote, heritable factors that are responsible for the inherited traits such as purple flowers or round seeds in pea plants. It is interesting to note that Mendel's publication came just 7 years after Darwin's 1859 publication of The Origin of Species, making the 1860s a banner decade in the development of modern biology. In his paper, Mendel stressed that heritable factors, which are today called genes, retain their individual identities generation after generation, no matter how they are mixed up or temporarily masked. Mendel probably chose to study the garden pea plants because they were easy to grow and they came in many readily distinguishable varieties. For example, one variety has purple flowers and another variety has white flowers. A heritable feature that varies among individuals, such as flower color, is called a character. A variant of a character, such as purple or white flowers, is called a trait. Perhaps the most important advantage of pea plants is an experimental model that Mendel could strictly control their reproduction. The petals of the pea flower almost completely enclose the egg and sperm-producing parts of the carpel and stamens, respectively. Consequently, in nature, pea plants usually self-fertilize because sperm-carrying pollen grains released from the stamens land on the tip of the egg-containing carpel of the same flower. Mendel could ensure self-fertilization by covering the flower with a small bag so that no pollen from another plant could reach the carpel. When he wanted to cross-fertilize, that is, the fertilization of one plant by pollen from a different plant, he pollinated the plants by hand. Thus, whether Mendel let a pea plant self-fertilize or cross-fertilized it with a known source of pollen, he could always be sure of the parentage of his new plants. Each of the characters Mendel chose to study, such as flower color, occurred in two distinct forms. Mendel worked with his plants until he was sure that he had a true breeding or purebred variety. That is, a variety for which self-fertilization produced offspring all identical to the parent. For instance, he identified a purple-flowered variety that, when self-fertilized, always produced offspring plants that had all purple flowers. Now, Mendel was ready to ask what would happen when he crossed true-breeding varieties with each other. For example, what offspring would result if the plants with purple flowers and the plants with the white flowers were cross-fertilized? In the language of breeders and geneticists, the offspring of two different true-breeding varieties are always called hybrids, and the cross-fertilization itself is referred to as a genetic cross. The parental plants are called the P generation, and the hybrid offspring are called the F1 generation. F stands for filial, which in Latin means son or daughter. When the F1 plants self-fertilize or fertilize each other, their offspring are the F2 generation. So this slide outlines Mendel's experiments, and we can see in the upper left corner the P generation where he hybridized true breeding plants. And then after the hybridization of the true breeding plants, this led to an all-hybrid progeny that has violet flowers. And then when he took the F1 generation and self-fertilized them, he noticed that there were purple flowers and white flowers in a specific ratio. Mendel reported his results of his crosses in a paper involving 7 different characteristics, each with 2 contrasting traits. A trait is defined as a variation in the physical appearance of a heritable characteristic. The characteristics included plant height, seed texture, seed color, flower color, pea pod size, pea pod color, and flower position. The characteristic of flower color, for example, the 2 contrasting traits were white versus violet or purple. To fully examine each characteristic, Mendel generated large numbers of the F1 and F2 plants, reporting the results from nearly 20,000 F2 plants alone, and his findings were consistent. But what were the results that Mendel found in his crosses for flower color? First, Mendel confirmed that he had plants that bred true for white or violet flower color. Regardless of how many generations Mendel examined, all self-crossed offspring of parents with white flowers had white flowers, and all self-crossed offspring of parents with violet flowers had violet flowers. In addition, Mendel confirmed that, other than flower color, the pea plants were physically identical. Once these validations were complete, Mendel applied the pollen from a plant with violet flowers to the stigma of a plant with white flowers. After gathering and sowing the seeds that resulted from this cross, Mendel found that 100% of the F1 hybrid generation had violet flowers. Conventional wisdom at this time in history would have predicted that the hybrid flowers would have been pale violet, or for the hybrid plants to have equal numbers of white and violet flowers. In other words, the contrasting parental traits were expected to blend in the offspring. Instead, Mendel's results demonstrated that the white flower trait in the F1 generation had completely disappeared. It is important to note that Mendel did not stop his experimentation there. He allowed the F1 plants to self-fertilize and found that, of the F2 generation plants, 705 had violet flowers and 224 had white flowers. This was a ratio of 3.15 violet flowers per 1 white flower, or approximately a 3-to-1 ratio. When Mendel transferred the pollen from a plant with violet flowers to the carpel of a plant with white flowers and vice versa, he obtained about the same 3-to-1 ratio regardless of which parent, male or female, contributed which trait. This is called a reciprocal cross. A reciprocal cross is a paired cross in which the respective traits of the male and female in one cross become the respective traits of the female and male in the other cross. For the other 6 characteristics that Mendel examined, the F1 and F2 generations behaved in the same way as they had for flower color. One of the 2 traits would disappear completely from the F1 generation, only to reappear in the F2 generation at a ratio of approximately 3-to-1. Upon compiling his results for many thousands of plants, Mendel concluded that the characteristics could be divided into expressed and latent traits. He called these respectively dominant and recessive traits. Dominant traits are those that are inherited unchanged in a hybridization. Recessive traits become latent or disappear in the offspring of a hybridization. The recessive trait does, however, reappear in the progeny of the hybrid offspring. An example of a dominant trait is the violet flower trait. For the same characteristic, that is flower color, white-colored flowers are a recessive trait. The fact that the recessive trait appeared in the F2 generation meant that the traits remained separate, not blended, in the plants of the F1 generation. Mendel also proposed that the plants possess 2 copies of the trait for the flower color characteristic, and that each parent transmitted one of its 2 copies to its offspring. Moreover, the physical observation of a dominant trait could mean the genetic composition of the organism included 2 dominant versions of the characteristic, or that included 1 dominant and 1 recessive version. Conversely, the observation of a recessive trait meant that the organism lacked any dominant versions of this characteristic. Moving on, the 7 characteristics that Mendel evaluated in this pea plants were each expressed as one of 2 versions or traits. The physical expression of characteristics is accomplished through the expression of genes carried on chromosomes. The genetic makeup of peas consists of 2 similar or homologous copies of each chromosome, one from each parent. Each pair of the homologous chromosomes has the same linear order of genes. In other words, peas are diploid organisms in that they have 2 copies of each chromosomes. The same is true for many other plants and virtually all animals. Diploid organisms utilize meiosis to produce haploid gametes, which contain 1 copy of each homologous chromosome that unite at fertilization to create a diploid zygote. For cases in which a single gene controls a single characteristic, a diploid organism has 2 genetic copies that may or may not encode the same version of that characteristic. Gene variants that arise by mutation and exist at the same relative locations on homologous chromosomes are called alleles. Mendel examined the inheritance of genes with just 2 allele forms, but it is common to encounter more than 2 alleles for any given gene in a population in nature. Geneticists distinguish between an organism's physical traits, called its phenotypes, such as purple or white flowers, and its genetic makeup, which is called a genotype. In other words, the 2 alleles for a given gene in a diploid organism are expressed and interact to produce physical characteristics. The observable traits expressed by an organism are referred to as its phenotype. An organism's underlying genetic makeup, consisting of both physically visible and non-expressed alleles, is called its genotype. Mendel's hybridization experiments demonstrate the difference between phenotype and genotype. When true breeding plants, in which one parent had yellow pods and one had green pods, were cross-fertilized, all of the F1 hybrid offspring had yellow pods. That is, the hybrid offspring were phenotypically identical to the true breeding parent with the yellow pods. However, we know that the allele donated by the parent with the green pods was not simply lost because it reappeared in some of the F2 offspring. Therefore, the F1 plants must have been genotypically different from the parent with the yellow pods. The parental generation plants that Mendel used in his experiments were each homozygous for the trait he was studying. That is, what made them quote-unquote true breeding. Diploid organisms that are homozygous at a given gene or locus have 2 identical alleles for that gene on their homologous chromosomes. Mendel's parental pea plants always bred true because both of the gametes produced carried the same trait. When the parental generation plants with contrasting traits were cross-fertilized, all of the offspring were heterozygous for the contrasting trait, meaning that their genotype reflected that they had different alleles for the gene being examined. Moving on, a cross between true breeding pea plants with purple flowers and a true breeding pea plant with white flowers is called a monohybrid cross. This is because the parent plants differ only in one character. Mendel saw that the F1 plants all had purple flowers. Was the heritable factor for white flowers now lost as a result of the cross? By mating the F1 plants with each other, Mendel found the answer to be no. Of the 929 F2 plants, about 3/4 or 705 had purple flowers, and 1/4, 224, had white flowers. That is, there were about 3 purple F2 plants for every white plant, or a 3-to-1 ratio of purple to white. Mendel concluded that the heritable factor for white flowers did not disappear in the F1 plants, but somehow was hidden or masked by the purple flower factor when it was present. He also deduced that the F1 plants must have carried 2 factors for the flower color character, one for purple and one for white. If we look at the figure on the right-hand side of this slide, we see the F2 generation. This diagram is of what we call a Punnett square. The Punnett square represents an equally probable product of fertilization, and that is outlined in the table at the bottom of this figure. This table contrasts the genotypic and phenotypic ratios. And as this table points out, it is impossible to distinguish a genotypic ratio by the phenotypic ratio in regards to the dominant allele. That is because where the dominant allele may be homozygous, it could also be heterozygous, but the dominant allele will always be the penetrating phenotype. So in order to know if an organism is heterozygous or homozygous for the dominant allele, a test cross may be done, and a test cross may determine whether an organism that has expressed a dominant trait was either heterozygous or homozygous for a particular allele in question. Moving on to the law of segregation, a sperm or egg carries only one allele for each inherited character because the two members of an allele pair segregate or separate from each other during the production of gametes. When a sperm and egg unite at fertilization, each contributes its alleles, and that restores the paired condition to the offspring. This equal segregation of alleles is the reason that we can apply the Punnett square to accurately predict the offspring of parents with known genotypes. Moving on, two other pea plant characteristics Mendel studied were seed shape and seed color. Mendel's seeds were either round or wrinkled in shape and either yellow or green in color. From tracking these characteristics, one at a time, in a monohybrid cross, Mendel knew that the allele for round shape, designated capital R, was dominant to the allele with wrinkled shape, designated lowercase r, and that the allele for yellow seed color, capital Y, was dominant to the allele for green seed color, lowercase y. But what would result from a dihybrid cross, that is, the crossing of parental varieties differing in two characters? Mendel crossed homozygous plants having round and yellow seeds with plants having wrinkled seeds. The union of dominant and recessive alleles from the P generation yielded hybrids heterozygous for both characteristics. In other words, it yielded dihybrids. As we would expect, all of these offsprings from the F1 generation had round yellow seeds, or in other words, the two dominant traits. But were the two characters transmitted from the parents to the offspring as a package, or was each character inherited independently of each other? The question was answered when Mendel allowed fertilization to occur among the F1 plants. If the genes for the two characters were inherited together, then the F1 hybrids would produce only the same two kinds of gametes that they received from their parents. In that case, the F2 generation would show a 3-to-1 phenotypic ratio, in other words, 3 plants with round yellow seeds for every 1 with wrinkled green seeds. If, however, the two seed characteristics sorted independently, then the F1 generation would produce 4 gamete genotypes, that is, capital R, capital Y, lowercase r, uppercase Y, uppercase R, lowercase y, and lowercase r, lowercase y in equal quantities. The Punnett square at the right of your slide shows all the possible combinations of alleles that can result in the F2 generation from the union of 4 kinds of sperm with 4 kinds of eggs. If you study the Punnett square, you'll see that it predicts 9 different genotypes in the F2 generation. These 9 genotypes will produce 4 different phenotypes in a ratio of 9 to 3 to 3 to 1. This Punnett square also reveals that a dihybrid cross is equivalent to 2 monohybrid crosses occurring simultaneously. From the 9 to 3 to 3 to 1 ratio, we can see that the ratio of plants with round seeds to those of wrinkled seeds is 12 to 4, as the ratio of yellow-seeded plants to green-seeded ones. These 12 to 4 ratios reduce to 3 to 1, which is the F2 ratio for a monohybrid cross. Mendel tried his 7p characteristics in various dihybrid combinations and always observed a 9:3 to 3:1 ratio, or two simultaneous 3:1 ratios, of phenotypes in the F2 generations. These results supported the hypothesis that each pair of allele assorts independently of the other pairs of alleles during gamete formation. In other words, the inheritance of one character has no effect on the inheritance of another character. This is called the law of independent assortment. Moving on, Mendel's two laws explain the inheritance in terms of discrete factors, or genes, that are passed along from generation to generation according to simple rules of probability. These laws are valid for all sexually reproducing organisms, including garden peas, Siberian huskies, and human beings. But just as the basic rules of musical harmony cannot account for all the rich sounds of a symphony, Mendel's laws stop short of explaining some patterns of genetic inheritance. In particular, they do not explain the many cases of inherited characters that exist in more than two clear-cut variants, such as flower color that can be red, white, or pink, or human skin color in all its ranges of shades. In fact, for most sexually reproducing organisms, cases where Mendel's rules can strictly account for the patterns of inheritance are relatively rare. More often, the observed inheritance patterns are more complex. For instance, when two genes are located in close proximity on the same chromosome, they are considered linked, and their alleles tend to be transmitted through meiosis together. As the distance between two genes increases, the probability of one or more crossovers between them increases, and the genes behave more like they are on separate chromosomes. We will now add several extensions to Mendel's laws that help account for this complexity. The F1 offspring of Mendel's pea crosses always look like one of the two parent plants. In such situations, the dominant allele has the same effect on the phenotype whether present in one or two copies, but for some characters, the F1 hybrids have an appearance in between the phenotypes of the two parents. This is an effect called incomplete dominance. For instance, when red snapdragons are crossed with white snapdragons, all the F1 hybrids have pink flowers, and in the F2 generation, the genotypic ratio and the phenotypic ratios are the same, which is 1 to 2 to 1. We also see examples of incomplete dominance in humans. One case involves a recessive allele that causes hypercholesterolemia. This is a condition characterized by dangerously high levels of cholesterol in the blood. Normal individuals are homozygous dominant. Heterozygotes have blood cholesterol levels about twice normal. They are unusually prone to cholesterol buildup in the artery walls and may have heart attacks from blocked arteries by their mid-30s. Hypercholesterolemia is even more serious in homozygous individuals for the recessive allele. These homozygotes have about 5 times the normal amount of blood cholesterol and may have heart attacks as early as age 2. If we look at the molecular basis for hypercholesterolemia, we can understand the intermediate phenotype of the heterozygotes. The dominant allele specifies a cell surface receptor protein that certain cells use to, quote-unquote, mop up excess low-density lipoproteins, or LDL, or bad cholesterol, from the blood. With only half as many receptors as the homozygous dominant individuals, the heterozygotes can move much less excess cholesterol. The ABO blood groups in humans is an example of multiple alleles and co-dominance. So far, we have discussed inheritance patterns involving only 2 alleles per gene, but most genes occur in more than 2 forms known as multiple alleles, although each individual carries, at most, 2 different alleles for a particular gene. In cases of multiple alleles, more than 2 possible alleles exist in a population. The ABO blood groups in humans are an example of multiple alleles. there are 3 common alleles for the character of ABO blood types, which in various combinations produce 4 phenotypes. A person's blood type may be A, B, AB, or O. These letters refer to 2 carbohydrates designated A and B that may be found on the surface of red blood cells. A person's red blood cells may be coated with carbohydrate A, giving them type A blood, or carbohydrate B, type B blood, or both for type AB blood, or neither for type O blood. Matching compatible blood groups is critical for safe blood transfusions. Our immune system produces blood proteins called antibodies that can bind specifically to the blood cell carbohydrates we lack. If a donor's blood cells have a carbohydrate that is foreign to the recipient's antibodies will cause the donated blood cells to clump together. This clumping can kill the recipient. The 4 blood groups result from various combinations of the 3 alleles. That is capital I hyperscript A for the ability to make substance B, capital I hyperscript B for the ability to make substance B, and lowercase I for neither A or B. Each person inherits one of the alleles from each parent. Because there are 3 alleles, there are 6 possible genotypes. Both the IA and IB alleles are dominant to the lowercase I allele. Thus, IA and IA lowercase I people have type A blood, and IB and IB lowercase I people have type B blood. Recessive homozygotes, which have 2 lowercase i's, have blood type O. They make neither the A nor the B carbohydrate. Finally, people of genotype IA, IB make both carbohydrates. In other words, IA and IB alleles exhibit codominance, meaning that both alleles are expressed in heterozygous individuals. Heterozygous individuals have type AB blood. Be careful to distinguish codominance, the expression of both alleles, from incomplete dominance, or the expression of one intermediate trait, as we discussed concerning the snapdragons. The patterns of inheritance we've discussed so far have involved genes located only on the autosomes, not on the sex chromosomes. We're now ready to look at the role of sex chromosomes in the inheritance patterns established by the characters they control. A number of organisms, including nearly all mammals, have a pair of sex chromosomes, designated X and Y, that determine an individual's sex. Individuals with one X chromosome and one Y chromosome are males. XX individuals are females. Human males and females both have 44 autosomes, that is, chromosomes other than the sex chromosomes. As a result of chromosome segregation during meiosis, each gamete contains one sex chromosome and one haploid set of autosomes. All eggs contain a single X chromosome. Of the sperm cells, half contain an X chromosome and half contain a Y chromosome. Therefore, an offspring's sex depends on whether the sperm cell that fertilizes the egg bears an X or a Y chromosome. Besides bearing genes that determine sex, the so-called sex chromosomes also contain genes for characters unrelated to maleness or femaleness. Any gene located on a sex chromosome is called a sex-linked gene. The X chromosome contains many more genes than the Y, therefore, most sex-linked genes are found on the X chromosome. A number of human conditions, including red-green color blindness, hemophilia, a type of muscular dystrophy, result from sex-linked recessive alleles. Red-green color blindness is a common sex-linked disorder characterized by a malfunction of light-sensitive cells in the eyes. It is actually a class of disorders involving several sex-linked genes. A person with normal color vision can see more than 150 colors. In contrast, someone with red-green color blindness can see fewer than 25. For some affected people, red hues appear gray, others see gray instead of green. Still others are green weak or red weak, tending to confuse shades of these colors. Because they are located on the sex chromosomes, sex-linked genes exhibit unusual inheritance patterns. For instance, when a colorblind male has offspring with the homozygous female with normal color vision, all the children have normal color vision, suggesting that the allele for wild type, in other words, normal vision color, is dominant. If a female carrier mates with a male who has normal color vision, the classic 3:1 phenotype ratio of normal color vision to color blindness appears among the children. However, there is a surprising twist. The colorblind trait shows up only in males. All the females have normal vision, while half of the males are colorblind and half are normal. This is because the gene involved in this inheritance pattern is located exclusively on the X chromosome, and there is no corresponding locus on the Y chromosome. Thus, females, XX, carry 2 copies of the gene for this character, while males, XY, carry only 1. Because the color blindness allele is recessive, a female will be colorblind only if she receives the recessive allele on both X chromosomes. For a male, however, a single copy of the recessive allele confers color blindness. For this reason, recessive sex-linked traits are expressed much more frequently in men than in women. For example, color blindness is about 20-fold more common among males than females. Hemophilia is also a sex-linked recessive trait with a long, well-documented history. Hemophiliacs bleed excessively when injured because they have inherited an abnormal allele for a factor involved in blood clotting. The most seriously affected individuals may bleed to death after relatively minor bruises or cuts. A high incidence of hemophilia has plagued the royal families of Europe. The first royal hemophiliac seems to have been a son of Queen Victoria, 1819 to 1901, of England. It is likely that the hemophilia allele arose from a mutation in one of the gametes of Victoria's mother or father, making Victoria a carrier of the deadly allele. Victoria later passed the allele on to her daughter and granddaughter, who were both carriers. Hemophilia was eventually introduced into the royal families of Prussia, Russia, and Spain through the marriages of 2 of Victoria's daughters who were carriers. In this way, the age-old practice of strengthening international alliances by marriage effectively spread hemophilia through the royal families of several nations. And that brings us to multiple alleles. We discussed multiple alleles briefly when we discussed the blood types. But Mendel implied that only 2 alleles, one dominant and one recessive, could exist for a given gene. Now we know that this is an oversimplification. Although individual humans and all diploid organisms can only have 2 alleles for a given gene, multiple alleles may exist at the population level such that many combinations of 2 alleles are observed. And as I said, we were discussing this concerning blood types. When many alleles exist for the same gene, the convention is to denote the most common phenotypic or genotypic trait among wild animals as the wild type. All other phenotypes or genotypes are considered variants. The variant may be recessive or dominant to the wild type allele. Moving on to pedigrees and diseases, Mendel's laws apply to the inheritance of many human traits. So if we call the dominant allele of any such gene capital A, the dominant phenotype results from either the homozygous genotype, capital A, or the heterozygous genotype, capital A, lowercase a. Recessive phenotypes always result from the homozygous genotype, lowercase a. In genetics, The word dominant does not imply that a phenotype is either normal or more common than a recessive phenotype. Wild-type traits, which are those most often seen in nature, are not necessarily specified by dominant alleles. In genetics, dominance means that a heterozygote carrying only a single copy of a dominant allele displays the dominant phenotype. By contrast, the phenotype of corresponding alleles is seen only in the homozygote. Recessive traits can be more common in the population than the dominant ones. For example, the absence of freckles is more common than their presence. So how do we know how particular human traits are inherited? Researchers working with pea plants or Labrador retrievers can perform a test cross, but geneticists who study humans obviously cannot control the mating patterns of their subjects. Instead, they must analyze the results of matings that have already occurred. First, the geneticist collects as much information as possible about a family's history for the trait. Then, the researcher assembles this information into a family tree or a family pedigree. You may associate pedigrees with purebred animals such as racehorses or champion dogs, but they can represent human matings as well. To analyze a pedigree, the geneticist uses Mendel's concept of dominant and recessive alleles and his law of segregation. Healthcare professionals have known for a long time that both common diseases and rare diseases can run in families. Family history can be used as a diagnostic tool and help guide decisions about genetic testing for the patient and at-risk family members. Moving on to polygenic inheritance, Mendel studied genetic characters that could be classified on an either-or basis, such as purple or white flower color. However, many characters, such as human skin color and height, vary along a continuum in a population. Many such features result from polygenic inheritance, the additive effects of two or more genes on a single phenotype character. Let's consider a hypothetical model of how polygenic inheritance might work. Assume that skin pigmentation in humans is controlled by 3 genes that are inherited separately, like Mendel's P genes. Actually, genetic evidence indicates that at least 3 genes control this character. The dark skin allele for each gene, capital A, capital B, and capital C, contributes 1 unit of darkness to the phenotype and is incompletely dominant to the other alleles, lowercase a, lowercase b, and lowercase c. So a person who is capital A, capital A, capital B, and capital C would be very dark, while an lowercase a, lowercase a, lowercase b, lowercase b, lowercase c individual would be very light. So the gist of it is the difference between persons with dominant alleles for skin pigment versus persons with recessive alleles in skin pigment produce contrasting skin colors. However, a person who is heterozygous for the skin pigment alleles, which would be capital A, capital B, lowercase b, and capital C, lowercase c, would have skin of an intermediate shade. Because the alleles have an additive effect, the genotype, capital A, capital B, lowercase b, capital C, lowercase c, would produce the same skin color as any other genotype with just 3 dark skin alleles, such as capital A, capital B, lowercase c, lowercase c. Moving on to continuous variation, most polygenic traits show the following characteristics. That is, continuous variation. Unlike Mendel's pea plants, humans do not come in 2 clear-cut, tall or short varieties. And complex inheritance patterns. In this case, many different patterns of inheritance are possible. For instance, tall parents can have a short child, short parents can have a tall child, and 2 parents of different heights may or may not have a child of intermediate height. We can also apply these principles to skin color, as I was illustrating on the previous slide. Moving on to the role of the environment. If we examine a real human population for the skin color phenotype, we would see more shades than just 7. The true range might be similar to the entire spectrum of color under a bell curve. In fact, no matter how carefully we characterize the genes for skin color, a purely genetic description will always be incomplete. This is because some intermediate shades of skin color result from the effects of environmental factors such as exposure to the sun. Many phenotypic characters result from a combination of heredity and environmental factors. For example, although a single tree is locked into its inherited genotype, its leaves vary in size, shape, and color depending on exposure to wind and sun and the tree's nutritional state. For humans, exercise alters build, experience improves performance on intelligence tests, and social and cultural factors can greatly affect appearance. As geneticists learn more and more about our genes, it's becoming clear that many human characters, such as a person's susceptibility to heart disease, cancer, alcoholism, and schizophrenia, are influenced by both genes and the environment. Whether human characters are more influenced by genes or by the environment, nurture or nature, is a very old and hotly contested issue. For some characters, such as ABO blood types, a given genotype mandates a very specific phenotype. In contrast, a person's blood count of red and white blood cells varies quite a bit depending on such factors as altitude, the customary level of physical activity, and the presence of infectious agents. Simply spending time with identical twins will convince anyone that environment and not just genes affect a person's traits. However, there is an important difference between these two sources of variation. Only genetic influences are inherited. Any effects of the environment are not passed on to the next generation. Characteristics that are influenced by environmental as well as genetic factors are called multifactorial. Moving on, the impact of a single gene on more than one character is called pleiotropy. An example of pleiotropy in humans is sickle cell disease, also sometimes called sickle cell anemia. This is a disorder characterized by a diverse set of symptoms. The direct effect of the sickle cell allele is to make red blood cells that produce abnormal hemoglobin proteins. These abnormal molecules tend to link together and crystallize, especially when the oxygen content of the blood is lower than usual because of high altitude, overexertion, or respiratory ailments. As the hemoglobin crystallizes, the normally disc-shaped red blood cells deform to a sickle shape with jagged edges. Sickled cells are destroyed rapidly by the body, and their destruction may cause anemia and general weakening of the body. Also, because of their angular shape, sickle cells do not flow smoothly in the blood and tend to accumulate and clog tiny blood vessels. Blood flow to body parts is reduced, resulting in multiple symptoms such as periodic fever, pain, and damage to various organs including the heart, the brain, and the kidneys. Blood transfusions and drugs may relieve some of the symptoms, but there is no cure, and sickle cell disease kills about 100,000 people in the world annually. In most cases, only people who are homozygous for the sickle cell allele have sickle cell disease. Heterozygotes have one sickle cell allele and one normal allele, are usually healthy, hence the disease is considered recessive. However, at the molecular level, the two alleles are actually codominant. Both alleles are expressed in heterozygous individuals, and their red blood cells contain both normal and abnormal hemoglobin. A simple blood test can distinguish homozygotes from heterozygotes. So let's recap tonight's lecture. After listening to this lecture, you should be able to explain why Mendel is considered the father of genetics. You should know the difference between a genotype and a phenotype. You should know what a test cross is, and you should know how to illustrate the law of segregation. You should be able to explain the law of independent assortment, describe incomplete dominance, know what a codominant trait is, know how you would identify a sex-linked trait, know what multiple allele traits are. You should be able to describe a pedigree and how they're used to track human diseases. You should be able to describe polygenic inheritance. You should be able to recognize a trait with continuous variation. You should know the impact that the environment has on traits. And you should be able to explain how pleiotropy impacts traits.